Starting with lipids. First of all, what are lipids? Heterogeneous group of organic compounds which are made up of carbon, hydrogen and few atoms of oxygen. Second property, they are water insoluble. But where are they soluble then? Soluble in non-polar organic solvents. Okay. When we categorize them, they can be simple fatty acids, simple fatty acid which can have or which has a carboxyl group. Where is it attached? This carboxyl group attached to an R group. That means the alkyl chain. Then the R group. What is this R group? It could be methyl or ethyl or a higher number of CH2 groups, alkyl groups. Okay, how many carbons should be there? 1 to 19 carbons at least. Then we categorize them into their types. These are of two types. Saturated fatty acids we say, and unsaturated fatty acids. In saturated we write butyric acid as an example, palmitic acid. In unsaturated ones we write linoleic acid, but the simplest lipid is glycerol. Glycerol is trihydroxypropane. Its structure is, this is the propane formula, right? Trihydroxy. Understood? Trihydroxypropane. So we call it glycerol. And a triglyceride is basically which is formed by the esterification of glycerol with fatty acids. Now moving to the second property of lipids, that lipids may be formed by esterification of glycerol. Glycerol is the simplest one, right? Trihydroxypropane. So its esterification is done with fatty acids. Fatty acid or fatty acids. So these can be monoglycerides, monoglyceride means with one fatty acid. It can be diglyceride. Diglycerides means two fatty acids. Or it can be triglycerides. Triglycerides means with three fatty acids. The lipids or the triglycerides, they are called fats, they are called oil. Why do they call fat and oil? That depends on their melting point. Alright. Now moving to the next property of its structure, a phospholipid. When we draw the structure of a phospholipid lecithin, we say first of all it is the primary basic chain CH2, CH and CH2. Here the H of this hydroxy group, it is replaced by O, C double bond O, R1. Okay. So what are we going to do is, we are going to replace this hydrogen with this entire structure. Again this side, when we have replaced, it will come O, C double bond O, R2, and here O, phosphate with the further complex derivative, and the structure is ready. We call it phospholipid lecithin. Okay. Now we are moving to our next topic, heterocyclic compounds. Hetero, as the name says, these are the organic compounds with heterocyclic rings, with two different rings. First are the nitrogenous bases. These are organic compounds with heterocyclic rings, and these are classified into two groups, purines and pyrimidines. Purines, these have two rings, and pyrimidines with the single ring. These are adenine, further divided into two types, adenine and guanine. And these are thymine, cytosine, and the third one is uracil. We are drawing the structure of adenine, NH2 group, and NH, here also N. This is adenine, heterocyclic, two rings, one is 6 carbon compound and the other one is 5 carbon compound. It's a purine. And the other one, pyrimidine, this is a single ring structure. This is uracil structure. Okay. Now moving to the B, nucleosides. First is the nitrogenous bases, second is the nucleoside. Nucleosides, organic compounds formed when pentose sugar, pentose is 5 carbon, pentose sugar, it can be, this pentose sugar can be ribose, can be deoxyribose, but it is carbon 5 sugar is attached to nitrogen base. So nucleosides are adenosine, guanosine, thymosin, cytidine and uridine. That means when the ribose sugar or the deoxyribose sugar is attached with the base. Base can be a purine and base can be a pyrimidine. Alright, this combination is nucleoside. And adenosine, guanosine, thymosin, cytidine, uridine are the examples if the bases are adenine, guanine, thymine, cytosine, cytidine as well as uracil. So, moving to the structure of adenosine. It's a 5 carbon compound. This is a 5 carbon compound where 1, 2, 3, 4 and the fifth one is here, CH2OH, fifth carbon. Alright, first carbon and the fifth carbon and the third carbon, these are the prime carbons which are responsible for its structure as well as for attachment, for bonding. Adenine. Adenine is the base which is present here. So this is adenosine. When we say uridine, it means OH, OH everywhere. Here the base uracil is present. Same way the fifth carbon is CH2OH. Okay, this is uridine structure, this is adenosine structure. Now what are nucleotides? Nucleotide means a nitrogenous base, pentose sugar, to which we recently call nucleoside. When has and attached to the another nitrogenous base and pentose sugar with the help of a phosphate bond, means it has a nitrogen base, it has a pentose sugar and a phosphate group, and basically we call them, these are phosphorylated nucleosides. That means a phosphate group is esterified to the pentose sugar of the nucleoside. Now here, how the name will change. This nucleotide will be termed adenylate acid. What? Adenylate acid. Why? Because it has nitrogenous base as adenine. It has ribose sugar and ribose or deoxyribose. We better write pentose sugar. And it has phosphate group. So we call it adenylate acid. Same we call guanylic acid. When we have the nitrogenous base as guanine, then cytidyllic acid, then thymidyllic acid, and uridylic acid. Here uridine, cytosine, thymidine, guanine. As per the nitrogenous base, so is the name of the acid. And what is the function performed by these nucleotides? These nucleotides, these are performing the following functions. First of all, they are the monomers of nucleic acids. Deoxyribonucleic acid or ribonucleic acid, they are the monomers which form the nucleic acids. Then higher nucleotides, with more than one phosphate group. When they have more than one phosphate group, basically store energy in their high energy phosphate bond. Then nicotinamide and riboflavin nucleotides. These function as coenzyme for oxidation process. We will do their function as coenzymes in further chapters. These basically function as coenzyme for dehydrogenases during the oxidizing process. That means the oxidizing enzymes. Then is primary and secondary metabolites. Moving to altogether a new topic. Primary and secondary metabolites. Now primary and secondary metabolites means the biomolecules which are basically found in living cells. Alright, biomolecules found in living cells. We call them metabolites. Primary. What do we mean by primary? Primary, which have identifiable features. They have identifiable function. They play a specific role. Where? In the normal physiological process. We can identify them, like we can easily identify amino acids, nitrogenous bases, proteins, nucleic acids. We easily know what is their role, what is their function performed, and how do we identify them. So primary, which are quite specific. When we move to secondary metabolites, products of certain metabolic pathways. These are the products of certain metabolic pathways which are useful to mankind, like rubber, drugs, spices, We do not know the basic nature. We generally know them. They are useful to us. These are the products of these metabolites. When these are specific, we call them primary. When these are converted methodologically into different products with different metabolic pathways and are made useful to us, we call them secondary metabolites. So further we are moving on to the higher units now. In the higher units, we are first talking about biomacromolecules. Biomacromolecules. That means the molecular weight in the range of 10,000 Dalton and above. Right? What is the molecular weight? 10,000 Dalton and above. Where are they basically found? In acid insoluble fraction. Obviously cannot be collected in the acid soluble part where the molecular weight is less than 20,000, 10,000. There are three types of macromolecules. First is polysaccharides. Then is number 2, nucleic acids. And the third one is proteins. Okay, what are basically all these? Polysaccharides, polymers of monosaccharides. Nucleic acid, polymers of amino acids. Proteins, nucleic acid, polymers of nucleotides. And proteins are polymers of amino acids. What are polymers? I am using a word polymer here. What are polymers? Polymers are further two types. They can be homopolymer or they can be heteropolymer. Homopolymer, only one type of monomers. Where the one unit is repeated n times. Okay? One unit is repeated n times. So we call it a homopolymer. Example is starch, inulin. And hetero is when two or more types of monomers are there. Example, proteins. There can be many in the chain. Now, starting with the first basic structure of proteins.